Welcome, everyone, to the BrainSpace Initiative. It's my pleasure to introduce Tor Wager. I've known Tor for many years because we teach an fMRI course together. I'll just give you kind of a brief intro of who Tor is. He's at Dartmouth College. He's the Diana L. Taylor Distinguished Professor there. He is directing the Cognitive and Affective Neuroscience Laboratory, which is focused on the neurophysiology of affective processes, pain, emotion, stress, and empathy. And he's been doing a lot of work in the use of neuroimaging and developing neuroimaging-based predictors and lots of other things. I think you'll enjoy his talk, and I look forward to it as well. And without further ado, Tor, I will let you take it from here. And thanks again for joining us today.
Well, thank you. It's really a pleasure to be here and speak with you. Glad I can do it, and excited you're here. All right, let's see. And let me know if you can't hear me okay. But human systems and human brain imaging has a unique role to play in a continuum from molecules to cells to systems to human health, in particular because it is a level of analysis that lets us link brain function directly to measures of human health and well-being. And to that end, I'm going to talk today about developing neuroimaging-based biomarkers, which are biological indicators related to health, disease, and that might respond to an intervention. And here they're going to be patterns of brain activity, any kind of brain model. And this can help us in a number of ways. They can serve as diagnostic biomarkers to help define a diagnosis that aligns with brain pathology. They can help to predict future disease progression. They can help us to predict who's going to respond to an intervention. And I think this is the most important category, is that they can serve as biological targets for interventions of all kinds. You know, if it's meditation or brain stimulation or pharmacotherapy, which systems are impacted in ways that produce benefits on human health. And another benefit of brain imaging in humans is that it provides this rich nomothetic net that links across many levels of analysis. So we can link patterns at the brain level to what's happening in animal systems, pharmacology, to genetics and genomics and increasingly large samples, to patterns of responses in our bodies, in our behavior, in our social networks, and of course to clinical diagnoses. So all this sort of sounds great, but the real question is, why haven't we gotten better measures out of fMRI that are clinically applicable? And this is a question that was raised by Tom Insall now about a decade ago when he was the head of NIMH. And I really took that and take that as a challenge to develop robust, reproducible, translatable measures into the clinic. Now, there's lots of reasons, sociological, political, economic, why this translation has been hard, but I'm going to deal with some of the scientific questions there, issues there. So the talk's going to have three parts. I'm going to talk first about predictive models and how we'd like to develop them. I'm going to talk about how we can use them to address some old ideas about affect and motivation and update those old psychological models. And finally, I'll talk a little bit about how we can use them to help understand interventions and how they work. So one starting point for this is a number of papers. Every few years, there's a paper that comes along that really critiques the field of functional imaging. It could be the voodoo correlations in social neuroscience paper, or a power failure paper, which criticizes small sample size. It could be about critiques of the way we do statistical inference, about the reliability of measures, about what they can tell us about mental function. One of my favorite debates, started by Max Coldhart, what can the brain actually tell us about the mind? Or maybe it's about all research findings in general. This is not a unique problem to the field of functional neuroimaging or human neuroscience or psychology by any means. It applies to animal neuroscience, but it really applies broadly across fields because there are some common principles of things that have gone wrong that we can address essentially by revising and updating the methods that we use. I'm going to give you my very, very brief summary of what these are. And again, not unique to functional brain imaging. but they operate here. And the first principle is that in standard mapping procedures, we have too many options. We have hundreds of thousands of brain voxels. Often they're interchangeable in terms of which voxel counts for an effect. There are many ways of pre-processing and treating the data. And all these options lead to a lack of rigor. It's hard to reproduce findings exactly. It's easy to overfit the data. Number two is too much noise. We have low sensitivity with brain imaging, which translates into low power and low reliability without very large samples in many cases. So the standard brain mapping approach, is really pretty terrible on this dimension. Number three, when you do find something, there are selection biases that operate. And so true effect sizes are much smaller than they appear. So it's very easy to find correlations in the literature between brain and behavior, or symptoms of 0.9 or something really strong. But the truth is, if you test that exact same marker again, the correlation is going to be much, much lower. And four, there's a lack of functional specificity. So there is no brain region that does one thing and one thing only. So if you look at activity one region at a time, there's too little information about mental states because there's so much overlap and convergence. So there's a lack of functional specificity. If we know we have activity in an area, it doesn't mean that we can say what the mental process is that was engaged. So a problem fundamentally is that the brain mapping approach doesn't match how the brain actually encodes information. I'm going to focus particularly on affect here, which is feelings, symptoms, motivation. And from many, many neuroscience studies now, the picture that's emerging is that the brain encodes affect in distributed patterns that are local neural ensembles. And those ensembles include intermixed populations of neurons that have different properties and respond to different things. So one of my favorite papers from Greg Corder and Greg Scher recently looks at neurons in the basolateral amygdala, and they identify a nociceptive ensemble that responds to painful things. But there's a lot of overlap. A lot of these neurons respond to other things as well in various patterns and combinations. So what I like here is that they tested mild touch and light touch and sucrose and shock and other aversive things as well. And so you can really see that there's this complex, overlap across these different ensembles. And third, these ensembles form networks that span many brain regions. So the amygdala alone has very little of the information compared to all of the other ensembles in different parts of the brain that it's connected with. Okay, so here they do find a lot of overlap, but they can tag and activate a nociceptor specific ensemble, and that affects pain behavior in a chronic pain model, but it doesn't transfer and change how they respond to chocolate or something like that, right? So you can get some specific behaviors. Okay, so as an axiom, the best model for capturing affect in the brain or any other outcome is the one that best matches the data generating process. So we're fairly far from that with the level of human imaging, but we can come as close as we can. And this idea that there are population codes or pattern codes that are distributed within regions and across regions has been one of the really dominant forces in understanding how neural function works. And so you can see all of these papers here in various domains across different sensory modalities document the importance of pattern codes, not just the sort of simple responses that are the strongest in any area. So as Bruno Averbeck and Alex Pouget wrote, as in any good democracy, individual neurons count for little. It's population activity that matters. So what we really need are neuromarkers that are predefined models that we test again and again in different individuals and samples and populations. We want them to match how those processes are encoded. So they should be based on distributed patterns and connections. We'd like them to be optimized to have good measurement properties. So high sensitivity, high specificity for particular categories of mental events, high reliability, replicability from sample to sample, and generalizability across different instances. So if we have a marker for pain, for example, we'd like it to respond to all kinds of pains of a particular class, or at least we have to know what the boundary conditions are. And to do that, we have to be able to test those models without changing the model in new individuals and new cohorts. So we're going to need to share the models as well. So this is what my lab has been doing in large part over the past decade or so. And the idea is that we'll take patterns of brain activity, and they could be linear patterns, which I actually prefer because they're the simplest to interpret and work with and cross-correlate with animal models. So as simple as possible, but not simpler. And they could be based on connectivity or based on graph properties. We've been really working on developing models of individual brain pathways that we can match more closely with animal models using some of the same techniques. And they can also have a computational modeling aspect that describes how the system dynamically evolves across time, making more precise predictions about its behavior at any given moment. And to the degree that we have such a model where we can characterize its measurement properties and we know something about what it measures, then it becomes interesting to test that model on various kinds of interventions. So if it's a model of pain or a model of fear or whatever, as I showed you, we can test how does it respond to a drug? How does it respond to a care context in which one's providing support for another person, receiving support from another person, or doing something like practicing mindfulness meditation? Does it actually have an effect on these systems or doesn't it? So we can look at the convergence and divergence across different treatments, and that's using them as a treatment target. So most of the studies, there have been now probably 1,000 papers that have basically used machine learning techniques to try to develop these pattern classifiers based on distributed patterns. And the vast majority of them use what I'll call the direct prediction approach, which is you have some brain features and you try to relate those features to clinical diagnostic categories like PTSD, for example, or depression. And some of the recent efforts have been done in larger and larger samples, which is great. They've been done in task-free measures, so mainly resting state fMRI and brain structure, which is not so great, as I'll show you. And the most common uses are to classify patients versus controls or predict clinical scores to find something that tracks a clinical outcome. Now, there's one of the latest in this long line of controversial papers is this paper in Nature by Merrick, Tarbo, Clemens et al. In this last year. And what's provocative about it is that they did a very thorough analysis of resting state and structural brain data in several large samples, the Human Connectome Project, ABCD study, and UK Biobank. And they try to develop univariate and multivariate predictions of many phenotypes, all kinds of different outcomes that you might be interested in. And the really important point here from all of these things they did is that the effect sizes are really, really tiny. So if you look at this histogram here of brain phenotype correlations, and this is from single regions at a time, the max correlation you get is somewhere around 0.1, and maybe it's even lower for measures of psychopathology. That's a really, really small effect. In fact, it's unacceptably low. It's to be sort of very useful in most cases. So statistical power is abysmal, really, until you have over 1,000 people or thousands of people. The probability of replicating a result. But even if you do replicate a result and you find something, these effects are way too small to be used as biomarkers for any sort of practical purpose. So that's the question is that really the best we can do? And one of the conclusions one might draw from this is that if you take direct prediction of outcomes in isolated regions one at a time from the resting brain, it's not going to work well. We've sort of maxed that one out. But it turns out effect size really matters. They also compared multivariate prediction of the types that I'll show you today, roughly speaking at least. So they have univariate on the x-axis here and multivariate on the y-axis. And what you notice from this line, each dot is a different phenotype they're predicting, like a trait or outcome, that the multivariate ones are about 4 times larger. And 4 times larger from 0.1 to 0.4 as a correlation, for example, is a big difference. In fact, you explain 16 times more variance and you need 16 times lower sample size for 80% power. So now what you could achieve, something you can detect with 80% power in an effect size, with effect size of 0.1, you need 3,000 people, With an effect size of 0.4, you only need a couple of hundred people for 80% power. So it really comes down to the effect size. And we actually published recently a response to this commentary, which was then responded to. I'm not going to spend a lot of time on it now, but what Thomas Spizak did is take essentially a pseudo-random sample of convenience of effects in the HCP study. He applied a standard algorithm that's a little bit better than the multivariate algorithm, so their effect sizes are a little bit larger, and calculated what you need for the sample sizes you need for 80% power. And for most of the time, so age is really good, actually. 100 people, you're going to be good, right? Some of these measures of cognitive ability and fluid intelligence are certainly pretty good, cognitive flexibility. Some measures like this task-based inhibition, which is not a reliable task really at all and relies on different scores. So it's not correlated with other cognitive tasks either. Then you need really large samples because the effect size is really small because the phenotype is not very good. So that's a drop on that. And there's much more to say, but that's a drop on that. So let me go back to this direct prediction approach. We have a review on this. Vince has a really great review on this too from the same year, I believe. And they kind of span the gamut. They run the gamut of different disorders. And most of them are diagnosis or classification of cases versus controls, but there are a variety of other types of biomarkers that have been looked at as well. And I want to make a couple of simple points about this. And one is that if you're trying to do this direct prediction between participants, what should the ceiling be? Well, it's limited by the heterogeneity of the disorder itself and by confounding factors. So let's say you take a population of high in psychopathology, could be PTSD, versus low, and you develop a classifier that detects the difference between them. Well, it turns out there are 636,000 different ways to meet DSM-5 criteria for PTSD. So you're not studying one disorder or a couple of subtypes or something. There's really a huge variation in that space. So your target isn't sort of one underlying process. That's a big problem. Secondly, you're comparing your high or low psychopathology group. There are many confounds. Those in psychopathology also are poorer in sleep. They use different medications. They use more drugs. They have more pain. They have more adipose tissue. They have more head movement in the scanner, which turned out to be quite a big difference. They can have complex effects, poor vascular health, blood flow, and other things as well. So when you're developing your classifier that detects PTSD versus control, it could be sensitive to some combination of all of these different things. And that's really a problem. So the approach that we've been taking over the past years is a different one. We call it a component process approach. It's a basic cognitive neuroscience approach. And the idea is we're going to emphasize within-person prediction in cases where we're trying to develop a brain measure that tracks a process that we can vary within a person over time with a task or a challenge. And then the idea is we'd like to find those things that can be encoded in a relatively straightforward way in the brain. And then we have measures of these underlying neurophysiological processes like pain, threat, fear of different types. And then those are combined in various ways to predict clinical and functional outcomes at the person level. So the hope is that this approach is going to be, work and bear fruit. Okay, so we're always then tracking within person variation as much as we can, at least. Each person's their own control. This is a result from one model, one of the first models we trained called the neurologic pain signature. It's one of the most widely validated. It tracks heat in response, or it tracks pain in response to heat, and you can just sort of see it responds during heat with a really nice response function that is tracks goes up with the stimulus intensity, predicts pain better than stimulus intensity, and doesn't respond to other types of the trial, other periods during the trial. And so I'll describe that marker now, even though it's a little bit old now. It's been around for a decade, but that means it's been tested in a lot of studies. So the really simple version of the idea here, and you can make it as complex as you want, is to take a training sample. Here we have 4 levels of noxious heat, from barely painful to as painful as people can tolerate. And we're training a giant penalized regression model that gives us a map, a signature map. And that has the weights that we can apply to data to best predict pain. And that map we called the neurologic pain signature because our journal editors wanted us to call it that, but it includes patterns in areas that we know are responsive to, they receive nociceptive input. They track pain. Some of them you can stimulate and affect pain in various ways. There's a rich animal literature on these regions. But it's not just that we have activity in these regions somewhere, right? It's that it is a fixed target, at least. When we test it, we're testing the same thing from subject to subject and sample to sample. So this is what happens when we apply it to a new sample and new individual test data. We take people, we take them one at a time. We can apply the signature to any images that we'd like, the time series, the condition averages. But for every image, it boils down this data image, this complex image, into a single response, which is the predicted pain. So that's our measure. And then we can empirically evaluate how well it tracks pain. So in all of the results here, the models are always tested in new individuals, out of sample, and many times with very diverse samples from different scanners. So whatever we're picking up here, that's evidence that there's a consistent brain representation across participants. We've done this as well, where everybody gets their own individualized model, and limited benefits to that, but that's a sort of a discussion point, right? There's various pros and cons, but I think it's important from a biomarker perspective that they're consistent. So this is 3 levels of noxious heat, 46, 47 to 48 degrees. Each line is a person. So the NPS response is going up across these levels of heat. And if we turn that into a classifier, a high versus low classifier, then we can use that continuous readout and say, can we predict which is more painful, the high or the low for a given person? And we would get it right about 96% of the time. And that's been our benchmark across studies. And now we've been testing this in a variety of different ways. I think it's really important to annotate that signature and keep testing it and keep pushing it and finding when it doesn't work and when it works. So this is a pain and placebo analgesia consortium, which involved at the time 20 studies, all of which involved pain of different types and placebo effects. And so this is a really wonderful way to test whether the NPS responds to diverse types of evoked pain in somewhat diverse populations, usually North American and European, although we've tested others as well. So we have 600 people with multiple types of pain: electrical, heat, laser, mechanical, visceral. And what you see here is each line is a study, and each dot is an individual person, participant. And the shift to the right means that the NPS is responding to painful stimulation across these different types. We would get the classification right for pain versus rest in 95% of the people. The average effect size is over 2 in this sample, which is much, much larger. Compare that to 0.4, right, with multivariate prediction in the resting state BWAS sample, for example. So what's different? Well, it's multivariate like the other one, but it's trained to track within person state, where each person serves as their own control and is compared to themselves. And we're not using rest or brain structure. We're using an appropriate functional task that's actually related to the pain state. And those things together is a game changer. So we have effect sizes that are 10 times larger than the BWAS results in the Merrick paper, where the max correlations you can get are about 0.1. Okay, we've done other things. We can look at its reliability as well. And this is the interclass correlation coefficient. It turns out Pain Report has a really high interclass correlation. It's very reliable. Excellent reliability, 0.91 here. And each of these dots is a different study, actually. So there's, I think, 9 studies in this original analysis, and we have many, many more details in the Neuroimage paper as well. But this is reliable. The NPS is close. It's a little less reliable, but it's pretty close. And some of these areas in these samples actually are quite reliable as well. So we, again, in a different sample of studies, have very large effect sizes for detecting pain versus rest, and also for correlations with pain within person. We get it right in almost every person. It's reliable. And what's interesting in this paper too is what we found is that even though both pain ratings and NPS are highly reliable, they're only modestly correlated. So one is not a proxy for the other. And you can think that's a bad thing, but actually I think what it really means is that the NPS isn't measuring pain in all situations and all types. And we've done a lot of work to unpack that. What it's measuring is a physiological system that contributes to pain in some people some of the time, particularly when the pain is delivered by a brief noxious stimulus that causes immediate pain in the body, as opposed to other kinds of things that people might call pain. So for example, if Vince, if I give him a heat pain on his arm, which I'd love to do next time we teach a course together, and then, and he tells me, bring it on, I don't feel anything, I'm really tough because he wants to impress me or doesn't want to give in, then that probably won't be reflected by the NPS, right? His low rating. Okay, so there's many things that it won't capture, including that kind of decision bias. We have been busy bees in some ways here. We've been working on these brain patterns for different types of affect. All of the things here, we have a brain pattern that's been tested with moderate to high effect size in cross-validated analyses, and also independently validated in a separate sample, which we haven't touched except for to test it once with the signature, and yields comparable effect sizes. So this is something that does work for many of these states. And the one take home, if you sort of take a broad view looking across all of these different things here, an interesting meta theme is that they're really quite different. I'm going to come back to that in a minute. So it's not as though all of the negative things group together and all the positive things group together. So that brings me to the second part of the talk, which is we've had ideas about affect and motivation for thousands of years since they're so important to us. And there are a few old ideas that I think need updating. And one of those ideas is this idea that there is an underlying fundamental distinction between positive and negative valence in the brain. So as Jeremy Bentham wrote many years ago, nature has placed mankind under the governance of two sovereign masters, pain and pleasure. They govern us in all we do and all we think. So I think it's sort of a beautiful quote. But, of course, we have to make decisions about the world. We have to decide what's good and bad. And so we have to integrate different kinds of painful things and different kinds of pleasurable things, so to speak, into some kind of action code, right? And how that happens, is an ongoing subject of debate. But the old idea is that we put these things on a bipolar continuum, and more pain is the opposite of pleasure, and vice versa, right? It turns out they might live in very different places in our brains, and the idea there then is that maybe different kinds of affective events, different kinds of unpleasant things, different kinds of painful things are really interchangeable. And what really matters is their valence. So that's the idea. And going along with that, these very early results, we found things like amygdala activity as a neural marker for threat or fear or negative emotion in general. And it's certainly been used that way quite often. But as Alfred North Whitehead said, seek simplicity and distrust it. So I think that's too simple to be the story. Why is it too simple? Well, we can take a meta-analytic view and we can start looking at studies of all kinds of different emotions: happy, disgust, fear, sadness, anger, and others. And here we've gathered studies that really kind of mainly are intended to elicit one kind of emotion category in people. We can take the findings from the studies and we can construct a Bayesian model. Jan Kang actually did this model, beautiful model, hierarchical Bayesian spatial point process model, and we can turn those study coordinates into an intensity function map and a full map of the joint distribution of activity across the brain for each of these different types. And when we do that, what we see, we look at the amygdala and we see that the amygdala is activated in all of these different emotion categories, first of all. So it's really not specific to any particular category like fear. And secondly, if we actually want to classify emotion categories and say what's systematically different about these different categories, we have to look at the marginal activity across the brain here. It's actually the patterns across all the areas that contribute, weakly, and their patterns of co-activation, which you can see represented in these graphs here. So there are different patterns of co-activation, even within the cortex, even within the thalamus, for example, in thalamocortical circuits, and the basic ganglia and so forth. So there are different patterns. When we apply those as a classifier to classify new studies, it does a surprisingly good job, which I'm really kind of shocked by. But you see this diagonal here that we get it right, 40 to 80% of the time in this analysis, which is not bad considering how coarse these data actually are. Okay, so that's about emotion categories. But I think we need to move then beyond the amygdala. One of the early attempts we used to do that is to take pictures like this spider here and other aversive pictures and neutral pictures, a range of different ones, and ask people, how negative do you feel? And when you do that, you can predict from the amygdala how negative they feel a little bit. You can explain some of the variation. Same with the anterior cingulate or the anterior insula. You can take a searchlight and say, what's the most predictive region across the entire brain? And that's biased, but it does better, although it is biased. It's a selection bias. And then you can take an unbiased whole brain predictive model, and that does a lot better at capturing variance in what people report feeling. And we did various analyses that show that you really do need the brain. You need multiple systems information from multiple systems together. So this model is a linear model, and what it does have activity in the amygdala, in the insula, in the anterior cingulate, but there are particular patterns. They're not things that we've named exactly. And those end up being important. Now, you can see some of the data here. When we apply this to in cross-validation, we get the light blue line. We apply it to a holdout data set, which we only tested once on the final model, one final model, and you get the same thing, which makes us believe that we're not fooling ourselves there. And you can turn that into a pretty accurate classifier test. Okay, here's another example. I'm going to show you two other models that seem like they ought to be similar, but they actually end up being somewhat different. So they're two threat-related predictive models. So in this one, Marianne Redden and Daniela Schiller and Daniela was studying threat conditioning and extinction. So what we have is a standard threat conditioning task where a tone predicts a shock. But there's a twist to it, right? So we're going to look at what happens during extinction. And the twist is that there's three groups in this intervention. So one group gets no extinction. So basically they get tone predicting shock, and then they're just tested later. A second group gets extinction. So they play the tone alone and the response to the shock goes down. In the third group, they just imagined the threat cue. They never actually saw it. So it's purely conceptual. Okay, so what we did is we trained a classifier there that tracks the threat cues. The CS plus is the threat cue versus the CS minus is a low threat cue. And we see a distributed pattern that does a pretty good job at classifying which is the threat cue and the non-threat cue. It includes parts of anterior cingulate and parts of the PAG, periocular to gray, as well as some signal in the amygdala. And then we can look and see how that pattern is expressed during extinction. This is what happens with no extinction. This is what happens with actual extinction. So the pattern response goes down with real extinction, which is great. What about imagined extinction? Well, it's also suppressed to the same degree here as in real extinction. So that's a neural target, right? We're using this as a target to see what happens when you have this innovative or different novel imagined extinction condition. The greater the signature expression, the greater the skin connections response. That's the standard outcome for threat tasks. And indeed, both real and imagined extinction also reduce SCR. So I want to contrast that with another kind of threat or fear signature. And this was done by Feng Zhu and Ben Becker and colleagues. And what they did is show people pictures specifically that elicited fear. So like this spider, for example, is one. They found a pattern also including increases in some of the same areas contribute to the pattern. They have some really nice validations that it responds when it's supposed to, and it tracks the subjective rating of fear on a 5-point scale, and a validation in a new sample. But the interesting thing about this is that these patterns and the pattern responses are different. They're not the same thing. So I showed you 3 patterns related to aversive picture ratings, conditioned threat, and unconditioned fear pictures. And those patterns do not make the same predictions. And they don't involve the same brain areas. So we have a multiplicity of systems that are probably tuned to particular kinds of stimuli and in different sensory modalities too. A second old idea that I think it's important to challenge is this idea that pain systems are the root of affective distress. So that when we describe the heartbreak of rejection as agonizing and thudding and dull and tearing and so forth, or searing pain in my heart, it's not just a metaphor. It actually evolved this capacity for social pain and vicarious pain evolved from our physical pain systems. And a large part of the basis for this is this idea that there's common activation across self-pain, other pain, and rejection, these social pain in the anterior cingulate and insula. So you get activity in the same areas in all of those tasks. But the problem is that you can start looking at what happens across many studies. So you can pick the most pain selective part of the anterior cingulate, which is this coordinate right there, and you can say, what are all the studies that activate that particular coordinate? And you can see there's a lot of things. So it doesn't mean that there's no nociception in that area. There are pain-related neurons. Pain's important, but it does so many different things that you can't look at that area and say, oh, it must have been the pain that was affected by any of these interventions. So, how do we separate then responses to physical and social pain? Does the NPS, for example, or pain signature respond to one or the other? What's it tracking? Is it tracking both? And so one way to address that is to manipulate things that are painful but not physically painful, like romantic rejection. And rejection is a great model because it happens early in life often. Its effects can last a lifetime, and they're very salient for us, and they're very aversive. And the question that we're going to ask here is a question about what's called separate modifiability. So in the anterior cingulate and across the brain, do we have overlap? Is a pain system responding to rejection? Or do we have separable systems where pain affects one system and rejection affects a different system, and neither one crosses over to affect the other? So in this study, we have 60 people who were all romantically rejected. They brought in pictures of an ex-partner and a friend, and they experienced painful and non-painful heat. So if Vince hobnobs with the elite, so maybe he brings in a picture of a close friend here and a picture of an ex, and he sees both in the scanner, and then he also experiences hot or warm heat in interleaved conditions. Both of these things evoke similar negative affect ratings, and they produce similar overall patterns of brain activity. In an earlier study that we did, we pointed out the similarities. But
well,
now,
I'm still thinking about this. I'm still thinking about the spider, so that's all I can do right now. So,
one of these things is going to get you, right? You should do a trigger warning on the spider. So there. So what happens with the NPS? Does it first respond to pain in this study? And it does. We can classify which is the hot versus the warm with 90% accuracy, so that's great. Now we can test the crucial condition. What happens when you see the rejector photo? And the answer is nothing different from chance. There's no rejector versus friend signal. And there's lots of ways in which we can test this at different moments with different levels of grief or rejection. But we looked pretty hard in this study. We didn't find anything that was like a pain signal. So this is what happens in the NPS when you actually get pain in red, or warm, low pain or non-painful warmth in orange, and then the rejector in the friend are even lower than non-painful warmth. Okay. So we can even look at the anterior cingulate itself. And this is the area where there's supposed to be the most convergence with pain affect for self and other romantic and physical. And what we can do is take that exact set of voxels and we can train a pattern that works, again, in out of sample participants to detect somatic pain versus warm, but it's that chance for rejection. And we can take this exact same set of voxels and we can train a rejection-related pattern that responds to the rejector photo, but it's that chance for whether you got painful heat or warm. So that pattern is separate modifiability, and that's strong evidence for process dissociation. So we've got two different processes happening in the anterior cingulate cortex here that are measured by two different patterns, and each responds to one and only one intervention. And normally, when we look at functional connectivity, we would take that region of the anterior cingulate and we would say, what's correlated with that region? That's a node. But what our claim is that there are separable patterns there that represent stable, across person, different processes. And so what we can then do is we can take these patterns as a seed in a separate resting state data set, and we can calculate for every person the pain-related DACC pattern and the rejection-related DACC pattern. So you do that for all the people, and then you ask what is functionally correlated or connected with each of those two different things, and are they different? And what we find is that they're radically different. The pain activates the sort of prototypical core pain systems, or connected to those other core pain systems. Rejection is connected to very different systems, especially like, for example, the lateral prefrontal cortex turns out to be quite important for predicting negative affect. Okay. We can do the same thing with watching other people in pain. So that's a pretty horrible thing. And I'm just going to jump to it. We have a sort of very carefully designed study where we have self-pain and other pain on two locations on the body with 3 intensities each. And we find that they're both really responsive. As the intensity of each of these things go up, then the pain ratings go up about the same for each condition. And we can look at the NPS and say, does it generalize from hand to foot? Yes, it does. So it works when it's somatic pain. What about the vicarious pain? Does it respond? It does not respond. In fact, it's deactivated overall. Okay. And we can actually then, we have the NPS, which responds only to somatic pain and tracks the intensity, and it also predicts the pain itself, the pain ratings. And then we can take the brains and develop another measure, what we call the vicarious pain signature, that tracks the intensity of perceived pain to others across these different levels, but it doesn't care at all about self-pain. So again, that's that separate modifiability criteria. And each one involves different systems in the brain. Okay, so the third idea that I want to talk to you about is this idea that there's a common neural currency for affective value. So they have to be integrated somewhere, right? But we have this idea that there are negative affect systems in the brain and positive affect systems in the brain, right? And so here we really set out to test that idea and update it. And so now we did this very ambitious study where we have 4 different kinds of affective stimulus. There's mechanical pain, that's pressure, heat pain, sounds, aversive sounds, like this knife on a bottle is really horrible, and pictures of other people in pain. And all of these things with 4 levels of intensity of each to create a lot of variation. All of them are aversive. We have people rate them on a common aversiveness scale. So they're all pretty aversive. They're not perfectly matched, but what we do is we tried to match them, and then we control for aversiveness too when we do our analyses. And the brain analysis here is a variant of the training that I showed you before, but it's a partial least squares variant. And the nice thing about this is that you can identify a multivariate pattern, but how you construct the outcome matrix, it's going to relate patterns of brain to patterns of behavior. And we constructed this outcome matrix here so that we could identify one target is a common pattern that that tracks aversiveness but doesn't care about what type of thing it was, and specific patterns that track only mechanical pain, for example, but don't care about the other types of things. And the idea about this PLS formulation helps us to separate those things. And again, all out of sample participants, and we've tested this in many other data sets as well in the paper. But the upshot of this is that there is a generalized negative aversive signature in this study. It's distributed, it involves many areas, and these are the responses here. So you can see it tracks subjective ratings across all the types, but doesn't really care much about which type of stimulus it was. It just cares about how bad it was. Okay, so in many of the sort of systems that we know and love, we have a very limited test of whether it responds to pleasant things as well. You know, where does it stop? Is it everything salient? But here it does track. We looked at these pleasant images. And it doesn't track the pleasantness of the images at all. And so we can identify some other systems that do that. So that's preliminary. It doesn't mean that there's nothing else that's arousing and pleasant that couldn't activate this pattern, but at least it's the start. And then the other interesting thing that we found from this is we can really look at where these things are encoded. And for this, long story short, we can look at the model weights with the bootstrap test and which model weights are really reliable. But also, we're going to take the conjunction of that with model encoding maps. So which brain voxels are really related to the overall model output. So just if you care about those things, it's the intersection between the bootstrap model weight maps and structure coefficient maps. And so then what that gives us is a small set of key areas. In this case, we're focusing on areas that are really stimulus type specific. So what we find is that with mechanical pain, we actually have areas in somatosensory cortex that care particularly about mechanical pain. And areas in the insula, anterior mid-insula, posterior insula, that care particularly about thermal pain. Areas in early auditory cortex that care particularly about auditory aversiveness. They respond in proportion to auditory aversiveness, and visual pathways, including visual cortex, early visual cortex, and amygdala, that really care a lot about how aversive a visual stimulus is. And the other interesting thing about this was that it includes these cortical areas, but these, in each of these cases, really extended to subcortical areas that are part of the sensory pathways. So the inferior colliculus, the auditory pathway, the inferior colliculus tracks aversiveness. And for vision, the lateral genicular nucleus, the visual thalamic nucleus, responds in proportion to the aversiveness. So we really think that the aversiveness itself is encoded in modality-specific ways that are specific to, that are really embedded in and entangled with sensory pathways. So we don't think that there's something that's general aversiveness. Well, we think there's a separate system that confers that, but we think there are these very modality-specific patterns. So this is what the data look like for these. So you can see that each thing tracks aversiveness in the condition that it's supposed to, and it really doesn't respond to or care about other things as well. And we basically replicated each of these different things in independent studies in the paper. So I think we tested 8 other studies in the paper. And if you want to know what makes somebody feel something's aversive, then we can look at whether it's a combination of this common pattern and modality-specific patterns. And it turns out to be a mix in both cases. So both are predictive independently. So we're not sort of reading off one fixed system, but a lot of the action here is not generalizable. It's different for different systems. Okay, so in the last couple minutes here, I'm just going to go back to this idea about understanding interventions. And one of the interventions I care the most about is placebo effects. What happens if you get a treatment that's not a treatment at all? It's just a sham drug. It's a manipulation of the context. And early studies had found that placebo treatment can reduce responses to painful events in seemingly pain-related areas, even reduce spinal cord responses to painful events, activate pain control systems, and cause the release of endogenous opioids. So this all points to sort of a deep sensory influence on how pain is constructed. So we can come back to our placebo meta-analysis, and we've already validated that it responds to painful events. Now, what about the placebo? And the answer is here, there's very little effect of the placebo, almost nothing. So the effect size and behavior is pretty large. People report less pain when they get a placebo. But here, the effect size is 0.07, so it's really small. It's statistically significant, but not much going on. So we have some choices to make. Well, that's I think, a marker for early nociceptive pain. But of course, there are many different kinds of things that can create pain in different ways. So we can do two different kinds of things. We can use the NPS or another similar signature as a target, and we can say which treatments have the largest effects on those nociceptive systems. So placebo, not so much. What about self-regulation? When you reappraise pain as burning, bubbling, sizzling, horrible, or spreading blanket on a warm blanket on a cold day. And so we train people and ask them to reappraise pain in this way. And we're going to ask, is it mediated by the NPS or not? And what we find is pain goes up and down systematically when people are reappraising up and down, so that works really well. What about the NPS? Well, it tracks the stimulus intensity of painful heat, but no response to reappraisal of pain. Okay, so that doesn't work. So placebo, no. Cognitive regulation, not so much. What about a state of mindful acceptance? Right. So here we train people to adopt a state of detachment, and really train them to not react to anything that's coming in. That's the accept condition, and that reduces negative affect to pain and to pictures. And here, when we test the NPS, what we find is there actually is a drop. There's a 25% drop. It's modest, but significant here. So that's really evidence that this works. The effect size here is larger than the average placebo effect size for sure, and just about any of the placebo effect sizes in 20 studies that we tested. What about receiving social support from another person during pain? So we had women come into the study and they hold their partner's hands or a squeeze ball while they're getting pain. And what you see here is NPS responds really beautifully during the pain, but not before and after. And the hand holding has an effect. It reduces responses during pain in this case. So that's an example of a second state-based manipulation that has an impact. And the last thing I'm gonna show you is this idea that there might be other pain link systems that are independent of nociceptive pathways. So this is exactly what we tried to do in this study. Across 180 people, we removed all the effects of stimulus intensity and the NPS, and we identified patterns that predict pain that's presumably more from endogenous sources, endogenous variations. And areas like the ventromedial prefrontal cortex and nucleus accumbens are really important here. And when we have people cognitively reappraise this spider, sorry again, Vince, but you might think this is a cute mama spider, so not so bad. And when you reappraise cognitively,
no, it's very bad.
not yet. What happens is you activate the VMPFC, activate the accumbens, and you feel better. Same with pain. When people cognitively regulated pain, they activate this pathway, and the more they activate it, the less pain they feel. We can apply the same thing to hand holding. So when people are hand holding, the NPS is reduced, but there's also reductions in all of these other areas as well. In the prefrontal cortices, in the amygdala, in the lateral frontal cortex, in the PAG, and somatosensory cortex. And then we can look for formal mediators of that, things that are reduced with hand holding and are correlated with reductions in pain report. And we see medial prefrontal cortex again, and orbitofrontal cortex, and other areas. So there's more going on than the NPS that's really linked to pain. Okay, and as a result, what we kind of have is, I've shown you two different kinds of systems. One is the NPS, which tracks nociceptive pain, and it is not influenced by many kinds of manipulations. And we have another sort of set of pathways, which is about conferring value and meaning. So medial prefrontal striatal systems. And we can look across studies at different manipulations. So the effect of social information, telling people that things are better or worse, affects the latter pathway, but not the NPS. Placebo works on this frontostriatal pathway. Imagined extinction has effects on this frontostriatal pathway. But some other conditions, like the right kind of conditioning procedures, can affect the NPS as well, or supportive touch, or mindful acceptance that I showed you. Okay, so we can use this to start to say which treatments are effective and which ones aren't. All right, that brings me to the end. I've talked about a lot of things about predictive models and how we can train them and what's gone wrong with the field. I've talked about 3 old ideas about affective motivation that have been around for a very long time, but that we can update in light of new neuroscientific evidence, and how we can use brain biomarkers to help us understand interventions and which interventions work in which way. So I'd just like to say thank you to my lab. There's a wonderful team of grad students and postdocs and collaborators, to collaborators and former lab members. Many of them are still working with us, and to our sources of funding who helped us do this work. And I'd like to say thank you for your attention.
Thank you, Tor. All right, so let me see. Armin, if you see questions, let me know. I'm trying to figure out how to bring these up.
Oh, wow, I see captions.
There we go. Okay, pipe in if there's any questions that I'm missing. I'm not able to see them here, but I had a question, Tor.
Yes.
Why the spider? No, okay, anyway. Burn me, stab me, anything. Just don't show me the spider. Okay, so in terms of the psychopathology, I was just wondering that 650,000 early intro slide you had, right, on combination. So that's of course, not actually the dimensionality, but I was just wondering what you thought the dimensionality of psychopathology was, or how could we figure that out, or how could we, so what are your thoughts there? It's a $1 million question, but yeah.
Yeah, it's certainly not that. I mean, I've just been lecturing on this too, and there's as you know, co-occurrence across conditions is so common, right? That's why the RDoC kind of exists, right? And we've been analyzing this in the UK Biobank, and I can't cite you chapter and verse, but we find remarkable co-occurrence across conditions, and across symptoms. So I don't know what the number would be, but I do think that there are some underlying processes like negative affect reactivity that are going to underlie many different disorders and be a risk factor. And I think that the kind of a cool thing about this approach is that I think that you could have these kinds of markers for that kind of process, and I think they're going to be expressed differently in different people in different conditions, right? So for somebody with PTSD, they're going to express that in response to like a trauma-evoked, trauma cues, right? But for somebody who's depressed, they might evoke that in response to self-related cues. So the dream is to sort of build a library of what some of those underlying, processes are in the brain.
Yeah. Cool. And I'll ask one more question, and then we'll see if others have some. But so obviously, I'm a big fan of the multivariate approach and sort of pattern-based, strategies for this. I guess I have a 2-part question, which is one, you were talking about sort of identifying these sort of patterns, like NPS, et cetera. But to what degree do you allow for or control for overlapping patterns? Right. So suppose you're trying to, this again gets at the issue of, confounds and comorbidity and things like that. So just because something is predictive of pain, for example, doesn't mean, could you improve that further if you had multiple overlapping patterns that were sort of separating out pattern-based signatures from one another?
Yeah, absolutely. Like using an ICA, for example.
That's a good one. No, but that's one of many. But yeah,
I think that the Checo paper that I showed you is like an attempt to kind of start to do that, right, with the PLS framework. But I think where we're moving and love to move is towards open-ended decoding. So we're gathering data from collaborators. We're forming a consortium. My grad student Mijin Kwan is working on this now, and we have about 3,500 different subjects, brain images from very, very different tasks, all kinds of affective tasks. And that, I think, will help us, provide the data set to do this. We need large, diverse data sets to do that, to do this sort of open-ended decoding, right, and disentangle some of these different factors. I think there's a long way to go there, but and that'll also kind of let us validate post hoc, right? Like if we have a pattern, we can validate which kinds of things is it sensitive to and which kinds of things is it confusable for that make it get it wrong, right? And then,
Yeah.
the refining the patterns, like the next level is this using these larger, more diverse data sets, I think,
Yeah.
if
Yeah,
it
and
works.
I'm only asking that because we find lots of cases where, again, that overlap is hiding information because you can separate things that cancel out if there are systematically non-overlapping patterns, but nodes overlap and they cancel within those nodes. So that kind of stuff, I think there's a lot to be done there still.
Yeah,
So
absolutely.
Lorenzo, So Lorenzo has a question. He says he has a question on the intersection of negative affect and acceptance. He wants to hear your opinion on whether patterns with varying degree of negative valence and acceptance could be reliably identifiable at the individual level and across sessions.
Yeah, that's a really great question. And wouldn't it be awesome if that were true, right? So, I mean, there's different parts to the question. One part is individualizing, right? So I think we're working on some stuff like this too. I think if you have very little data per person, individualizing, you don't going to get a very good reliable signal. So sometimes the group patterns do even better than the individuals. But I think with a lot of data per person, there's obviously a lot of individual differences in the brain basis of these things. And we, published some of it, like the pain systems, there are some of these areas really are more different across individuals, right? So there's a lot of value in sort of identifying subtypes and customizing them. So I think that's really cool. You need a lot of data per person. And when you go to the acceptance thing, I think that would be really great if that worked. So you have to vary the sort of bottom up affective challenge and vary the degree to which you're engaging in a process like acceptance. We have a paper that we just finished the draft of that really is about emotion regulation. We think that we can detect cognitive reappraisal demand quite accurately, and we're moving closer to being able to sort of identify a system that really detects the degree of regulatory capacity and predicts regulation success. We haven't tried with acceptance. I think that would be really interesting. And the question is, can you do something that captures the variation in that state, right? So yeah, it's open field.
Yeah. Great. Yeah, he says thank you. Okay, so any other questions for Tor?
Yes. I unmute Eddie for you to ask your question.
Can you hear me?
Okay.
Yeah, I can.
Yes.
Hi, Tor. I'm Eddie Namias. I'm in philosophy and neuroscience down here. I've published with Adina up there, who I assume you know.
I do. Great. Hour this morning.
Okay, that was really fascinating, and there's a lot to be asked about. But I'll focus on one thing you just really quickly mentioned, which was the inhibitory response to observing pain in others relative to feeling pain in yourself. And I just don't know enough to know whether that's generalizable to other sorts of self-other activities in the brain, and also more speculatively whether that there might be some way to use that to intervene on pain, by getting people to sort of dissociate from their own bodies and perceive what's happening to them as if it were happening to someone else. That might be what's happening in mindfulness, or even some placebo effects. But anyway, it's a really cool effect, and I didn't know if it happens all over the place.
Yeah, I mean, so I just quickly to the last part first. I think that this idea of distancing or dissociation or disidentification is really an interesting and kind of powerful idea. So I think it's really possible that there's like a lot of mileage there, right? That we can take advantage of more systematically, right? And I think to the first part of this question, I have a sort of I'll tell you what I think is happening there, right? So with the NPS, you deactivate it because I think what's happening is that a lot of the affective value related signals are embedded in sensory cortex. And so if you're getting pain, the more pain you're actually deactivating some of these sensory systems. And when you get aversive pictures or aversive sounds, you're activating these sensory systems in proportion, and you're relatively deactivating the pain, pain-related regions. So they're really pretty different. I think what's happening, with the NPS is that there are these regions that have negative weights in the NPS that are where lower activity is predicting more pain, and they are some of these like high-level visual areas. And those are exactly the areas that you activate when you're watching somebody else in pain. So you get a net decrease, right? If you actually take only the positive nociceptive parts of the NPS, you get 0 response for the vicarious pain. You don't get the deactivation. So I don't know how much that sort of made sense or didn't make sense, but I think it's because they're really tracking systems that are competing at some level, right? And so yeah, so even though both are bad things, right? And I think it goes along with the story that that a lot of the aversiveness actually is in modality specific and task specific pathways, rather than sort of being one thing across the brain.
Great. When you say deactivate, Tor, are you talking about actual deactivation of neurons, or are you talking about decreases of fMRI signal?
I'm talking about always decreases in fMRI signal, right? And then
Okay.
Complicated relationship at the neural level, but yeah. Right.
Yes, as we know, yes. So just quibbling on that, yeah.
Yeah,
all right, cool.
Yeah, that's right. Good.
Well, we are at 2 o'clock, and I don't think. Oh, here we have. Yeah. Okay. So good. So I think we're good to go. So Tor,
right.
Yeah. So Tor, for the just to let you know, for the Brain Space Initiative, we are recording this, and we'll have a Slack channel devoted to your talk, and so there may be some follow-up this time, sort of opportunity for people to maybe ask some questions and interact with one another as well as you potentially. So hopefully this is for me, this is really good to see you again, and very interesting talk, and thanks again for joining us.